Hi everyone, this is Dr. Asi. Welcome back to the channel. So today's topic is cell photosynthesis and cellular respiration. Just reviewing these three chapters. So we'll start with first we will start with cellular respiration. Let's get started with cellular respiration. Okay, so cellular respiration and fermentation, very important chapters. So you see just a summary of cellular respiration here, which is energy yielding reactions. And here is its formula, C6H12O6 plus 6 oxygen, which yields 6 CO2, 6 water, and about 32 ATP. So let's get started. Okay, so this is a catabolic reaction. It means it yields energy by oxidizing organic fuels. So it starts with glycolysis, which is oxidizing glucose to pyruvate. Then after pyruvate oxidized, citric acid cycle completes the energy yielding oxidation of organic molecules. During oxidative phosphorylation, which is the last part, chemiosmosis couples with ETC, the electron transport chain, to ATP synthesis. And one alternative to this whole thing is after glycolysis, it could, if there is no oxygen available, it could go make a left turn from there and actually go fermentation, which is producing ATP without oxygen. Only some organisms can do that. So, well, all living cells, they need energy. They require energy from outside sources. So some animals, they can obtain it from plants like giraffe. Some animals feed on others. Again, they use catabolic pathways to yield energy from fuels, organic fuels, right? So fermentation, which is basically is actually done without oxygen, anaerobic. So cellular respiration, it must have oxygen. Okay, so and this is a redox reaction. So in a redox reaction, something must be oxidized, something must be reduced. So in this case, the glucose, our starting molecule, oxidized to CO2, and oxygen, our second molecule here, reactant, is reduced to H2O, right? So there are many steps. There is a molecule called NAD, which is or NAD plus, and then this is a molecule with this like a coin with two sides. NAD one side, the other side is NADH. So according to a lot of studies, you need to have more NAD, NADH, higher ratio. So we don't use NADs and convert them NADH unless eat food and then there is food available and NADH, convert the NADH to carry this food. But eventually NADH really carries them, carries these electrons actually. So NAD and NADH, they go together. So if you are not eating, so you have more NAD. If you're exercising, you decrease the NADH. Okay, so NAD is an electron. NAD is an electron acceptor. NADH is the reduced form of same molecule which carries electrons. Okay, so cellular respiration uses NAD and NADH, convert them NADH and carry stuff, passes the electrons to ETC, electron transport chain, and oxygen there actually pulls the electron down in the chain and then yielded is used, energy yielded used to regenerate ATP eventually. So glycolysis, citric acid cycle, oxidative phosphorylation, three big parts. And each part, if you collect all the ATPs, we have up to 32 ATP per glucose molecule. We can actually create from a single glucose molecule. Okay, so glycolysis, make sure you study these 10 steps of glycolysis. After pyruvate oxidized, citric acid cycle completes the energy yielding oxidation of organic molecules, but it must be in the presence of oxygen. So, that goes to the citric acid cycle, which is also called Krebs cycle. So same thing, okay? So there are 8 steps in the citric acid cycle, which takes the acetyl-CoA and then makes 2 turns, and then creates 2 ATP, and also net yield of 6 NADH, 2 FADH2, and CO2 from citric acid cycle. So again, 2 terms. The next is ETC, which is electron transport chain, which happens in inner membrane of mitochondria, which is crystal, so the location. So ATP synthase enzyme is also there, so which also helps this. So this chemiosmosis. So ETC plus chemiosmosis, again in crystal, is basically all together. They work together here to create maximum number of ATP. Okay, so with the ATP synthase enzyme protein, they use oxidative phosphorylation. So ETC plus chemiosmosis, oxidative phosphorylation. Okay, so total ATP is 30 to 32. Fermentation is an alternative. It could be alcohol fermentation or lactic acid fermentation. So they use substrate level phosphorylation. Net ATP is only 2 from fermentation, but it's used in a lot of industry actually to create so many food products using this fermentation. Okay, so also don't forget proteins, carbohydrates, and fats all can be actually used and get in this reaction from different places to actually create ATP. So always remember that. And this is also regulated using feedback mechanism. If ATP levels drop down, it needs to be speed up, and vice versa. If you have too much ATP, it needs to be inhibited to lower this speed. So that's cellular respiration. Next chapter is photosynthesis. So photosynthesis is basically photosynthesis is the most important reaction in biosphere. So we know that the formula is 6 CO2 plus 6 water, sunlight, chlorophyll, and then products are C6H2, H12O6, 6 oxygen. Now, so we must have water, CO2, sunlight, or light that actually, and chlorophyll to start photosynthesis. Okay, so photosynthesis feeds biosphere. The light reactions, so there are 2 major groups of photosynthetic reactions. It's light reactions, first part, Calvin cycle, second part. Light reactions convert solar energy to chemical energy of ATP and NADPH. Calvin cycle reduce CO2 to sugar. And there are 3 major types of alternative carbon fixation mechanisms of photosynthesis. They're called C3, C4, and CAM. So again, photosynthesis feeds the biosphere. The photosynthetic organisms, they are also called autotrophs. They are producers, like plants, cyanobacteria, some unicellular eukaryotes, and the purple sulfur bacteria and algae. So my multicellular algae, they are all they can all carry out photosynthesis. In plants, leaves are the main location where chlorophyll is located, especially mesophyll cells. Inside there, we have inside the cell, we know chloroplast is the organelle. Inside chloroplast, thylakoids, where actually light reactions start. So 2 stages of photosynthesis, light reactions versus Calvin cycle. Light reactions happen again in thylakoids. They can split water to release oxygen, reduce NADP plus to NADPH, and generate ATP. But always remember, ATP generated here is not an available ATP. It's actually used in Calvin cycle. So we need ATP and NADPH from here to move to Calvin cycle. Oxygen can leave the reactions and available. Okay, so Calvin cycle, named after Dr. Melvin Calvin, a Nobel laureate, who actually came with this model, Calvin cycle. It's actually we need CO2 to start the Calvin cycle. and we need an enzyme like Rubisco if you are talking about C3 photosynthesis, or you might need PEP carboxylase if you are talking about C4 photosynthesis. Calvin cycle uses chemical energy of ATP and NADPH to reduce CO2 to sugars. Okay, so the sugar that generated from Calvin cycle is called G3P, glyceraldehyde triphosphate. Later, that sugar converted to 1 glucose per 2 G3P. So every G3P can make 1 glucose, and they can. So that's glucose after that. Okay, so that's Calvin's cycle. Photorespiration using oxygen instead of CO2 to produce CO2. If this happens, if there is not enough CO2. So again, I mentioned there are 3 types of photosynthesis, C3, C4, and CAM. So default one is C3 photosynthesis, which is the cool season plants like wheat can do that. But there is another photosynthesis type called C4, like corn, more warmer season plants. And it can be very efficient photosynthesis. C4 in corn, sorghum, they use more efficient CO2 gathering, and they also minimize photorespiration. So, and they are better in warmer climates. And the last one is crustless and acid metabolism, also called chem photosynthesis, like agave, yucca, cacti, or pineapple. So they use this night and daytime, like 2 levels of different timing photosynthesis. Again, only 5,000 species can do that. They close their stomata during the day and only open it during the night. So that's photosynthesis. And our last chapter is cells and membranes. Okay, so we know that cell, so this is the smallest unit of every organism, and it's very, very important. In order to study cells, we must use a microscope because they are just too small, and we need to pay attention to this animal cell versus plant cell, and make sure that you actually know all the organelles, also plasma membrane, so that and be familiar with. So there is something called endomembrane system in the cell. There are 6 organelles that they are linked to each other. Nuclear envelope, Golgi, lysosome, vacuoles, plasma membrane. They make endomembrane system, and then they communicate using vesicles between them. So can be smooth or rough Smooth means they don't have ribosome. Rough have ribosomes. So if they are smooth, they can actually synthesize lipids, metabolizes carbohydrates, store calcium. If they are rough they actually obviously make proteins, distribute, transport vesicles. They are the membrane factory of the cells. Golgi can ship the material, receives and ship. Lysosome can actually have digestive compartments using hydrolytic enzymes. Vacuoles can be food vacuole formed by phagocytosis. Contractile vacuoles found in freshwater or protists pump excess water out of cells, and central vacuoles, which is found in mostly plant cells. Mitochondria and chloroplast we just seen in last 2 chapters, so which is cellular respiration and photosynthesis takes place. So they exchange energy, mitochondria and chloroplast. They have other things in common. They also both have DNA, and they can actually have genes in them, chromosomes and genes. So they are both very, very important organelles. Okay, chloroplast found in plant and algae. They are sites of photosynthesis. Again, they both have double membranes. They contain free ribosomes. Circular DNA molecules grow and reproduce independently in the cells. According to the endosymbiont theory, an early ancestor of eukaryotes engulfed an oxygen using non-photosynthetic prokaryotic cells and formed relationship between host cells and becoming an endosymbiont and evolved into the mitochondria. So mitochondria is the energy powerhouse, which has matrix and intermembrane space. and again has DNA in it too. Chloroplast we talked about, thylakoids, cytoskeleton, which is supports and cells and maintain its shapes as motor proteins. It could be microtubules, microfilaments, and intermediate filaments, 3 types. Cell walls are the extracellular structure, especially in plant cells and fungi cells as well, even though their material is made of is different. One is cellulose, the other one is chitin, but they both have wall. The wall can be primary wall, middle lamella, which is like a glue, holds everything together, or secondary cell wall. And the plasmadesmata is the little tiny holes on the wall. Animal cells don't have wall, but they can have ECM, which is extracellular matrix, which is outside the plasma membrane, made of glycoproteins like collagen, proteoglycans, fibroglycans, and then ECM proteins bind to receptor proteins in plasma membrane called integrins. So that brings us to membrane. So let's talk about membranes a little bit. They are fluid mosaic of lipids and proteins, like plasma membrane, and they are selectively permeable, and they can have passive transport like no energy, or active transport using ATP, or bulk transport like exocytosis or endocytosis. Again, plasma membrane is selectively permeable. Only some things can easily pass, some other things cannot, and has fluid mosaic models, which is proteins, carbohydrates, steroid cholesterols. They all actually in this mosaic, in a double, like a sandwich model. Okay, membrane proteins, they can be peripheral, like on the surface, or integral, going from one side to another side. They're also called transmembrane proteins. So membrane proteins can have one of these 6 functions, like transport, enzyme, signal transduction, cell-cell recognition, intercellular joining, or attaching cytoskeleton to ECM, like in animal cell. So synthesis, you can read about how they are synthesized. It's a double bilayer of lipid. Always remember there is hydrophobic sites like hydrocarbons, and they can easily dissolve and pass, but hydrophilic molecules cannot pass easily to the membrane. They need some type of transporter proteins to help them. It could be used as a passive transport like diffusion or osmosis. Osmosis is just diffusion of water from high water to low water. They could be isotonic when they reach same level. It could be hypertonic. Outside is less water, high solid, or hypo, HYPO, tonic, then the opposite. The outside is more water. Diffusion, active transport actually uses ATP in a pump, like sodium potassium pump. Then it can move opposite direction, like low to high, no problem. And make sure check the membrane potential, and co-transport, bulk transport. And this is the end of cell chapter. So I'll stop here. This is it for today, and good luck with the studying these chapters. I will see you in next video. Thank you for watching, and please subscribe to the channel. Bye everyone.